Hi guys, so this is the second part of chapter 28 starting at fertilization and then chapter 29 embryology all in one. Okay, so let's get started. Fertilization. We have a picture here of the sperm, which is the smallest cell in the human body, and then an egg, which would be the secondary oocyte here. So remember that with each ejaculation you have about 5 million waiting in the epididymis to go at any time, and they get dumped into the vas efferents, and we'll get to all that here in a little bit. So the first sperm, we'll say, let me get my yellow out. We have about 200 to 500 million and of these 200 to 500 million sperm, about 100 actually make it to the egg. So in reality, you only have to have about 70% of those 500 million be healthy. So be healthy to be fertile. And then of those 70%, only 100 will actually make it all the way through to the egg. And then only one actually makes it into the egg. The first sperm that actually gets in binds to a receptor on the zona, within the zona pellucida. So let's draw that here. Here's our zona pellucida. And surrounding it is, remember, our ring of granulosa cells called the corona radiata. So that's the corona radiata. And let's look at our sperm here. What does our sperm actually look like? It is the smallest cell in the human body. And here at the head, we have the nucleus, which takes up the majority of the head. And remember, it's full of DNA. Okay, and at the tip, we have this section right here called the acrosome. That is full of digestive enzymes that will actually act on the egg to digest these protective layers that are surrounding the secondary oocyte. And then, of course, you have here the tail. Okay. So the first sperm that gets in will bind to the receptors on the zona pellucida. So in reference, the sperm is much smaller than the egg. So you have 100 of these little sperms that actually make it to the egg. And the first one to make it to the receptor actually then locks down all the rest of the receptors, locks down the egg to all the other sperm. So then no other sperm can actually enter the egg itself. And then finally, the secondary oocyte can finish going through meiosis. And what you will produce is a zygote. So for like 2 seconds, so now our sperm has entered in. So here is our, we'll do it in blue, the nucleus of the sperm. So very briefly here, now we have an ovum, and this will shoot out a polar body, which is just, remember, a blob of extra DNA that it gets rid of. And it finishes going through meiosis. It's very briefly an ovum. And now we've actually made a baby, and we've formed a zygote. It's really the very first cell that is going to become you. And then the zygote will actually go through mitosis. Okay? It's bouncing around, the zygote is bouncing down the fallopian tube towards the uterus for about 4 to 5 days. The fallopian tube makes its own food for the zygote. It's actually called fallopian milk. And then it is going to reach the uterus, and it floats for about 5 days till it finds a spot to land and then digs in and really just eats the lining of the uterus. And the uterus makes its own milk called uterine milk and feeds the embryo. So let me see what the other page is. Okay. So, like before, now we have a zygote. Here's our zygote. It's going through mitosis. It bounces down the fallopian tube and heads to the uterus for about 4 to 5 days. It reaches the uterus and floats for about 5 days till it finds the spots and digs in. The zygote actually produces a hormone called HCG, human chorionic gonadotropin. Human chorionic gonadotropin. So let me write that out for you. And something interesting, it actually tells the corpus luteum here to stay alive. If you remember, corpus luteum only stays alive for about 12 days. And here, that HCG, or that zygote actually, is going to tell, the zygote that's making the HCG is going to tell the corpus luteum to stay alive. Okay, and it will stay alive for about 7 weeks, and then something else will take over the progesterone production. Okay. Also, what else does HCG do? HCG will also cause a boy fetus to make testosterone. So if you remember, when we start out, the zygote starts out, we start out with what's called bipotential gonads. Bipotential gonads, meaning they can go female or they can go male. And what this HCG does is that if it's a male, then it will cause it to produce testosterone to become male gonads, so to produce testes. So if it has a Y chromosome, then it will produce testosterone. The HCG will cause it to produce testosterone and then produce testes to become male. Okay. I think I told you everything on this. All right. So let's sort of travel back and talk about what happens in the male. How we actually get the sperm inside the vagina. So if you remember, we have about 500 million sperm waiting in the epididymis for go time. And then they get dumped into the vas deferens. And remember, this is in the spermatic cord. Okay. We have some other guys that play a role as the spermatic cord. This is the spermatic cord here. It comes up and goes over the bladder here. Here's the ureter. The spermatic cord comes down, containing the vas deferens, And then we have this gland. And that's the seminal vesicle. And the seminal vesicle actually donates some stuff to the sperm as they are moving along here. It donates sugar, so a little snack for the sperm. It donates bicarb. So if you remember, the environment inside the vagina is very acidic, and the sperm don't like that, so they need something to combat that, and that's what the bicarb does. It also donates what's called prostaglandin, which is a hormone. And what it acts to do is actually soften the cervix. So you may have heard that when a woman is close to pregnant or close to giving birth, that having intercourse will actually help to induce breaking the water or going into labor. And that's actually because of this hormone right here. It actually acts to soften the cervix and cause the cervix to soften and for her water to break. All right, so the seminal vesicle adds its mix to the sperm. It joins here. Remember, we're in the vas deferens, and as it joins with the seminal vesicle, it becomes the ejaculatory duct. And then it starts to pass through another gland. And that's the prostate. Okay, so that's here. And the prostate also donates some stuff. It donates food like sugar, so another snack for the sperm. And then it donates antibiotics. And then what's called PSA. I think it stands for prostate stimulating agent, which is actually a semen liquefier. So before, the semen is somewhat gelatinous, and it's really hard for the sperm to swim through that. And so this PSA makes it more liquidy and allows the sperm to swim easier. So as it travels through this prostatic urethra, it enters this small section right here called the membranous urethra. And then another gland adds to it called the bulba urethra gland. And what the bulba urethra gland adds to the mix is mucus. It's the final addition to the semen, and it makes the semen very slippery. So it allows the sperm to come on in and be able to swim very easily and make it into the cervix and up through the fallopian tube. And then finally, it makes its way into the penis through the spongy urethra. And then if it's lucky, it will enter the vagina. And then up into the cervix, which is the hole at the end of the vagina, or at the end of the uterus. And then into the uterus, where the sperm will pick a side, either right or left. Half will go one way, half will go the other. And then up into the fallopian tube. And the lucky ones will find the egg. Okay. So here we are back at this picture. The corpus luteum, the embryo that was a zygote, is producing HCG, telling the corpus luteum to stay alive. So HCG keeps the corpus luteum alive for about 7 weeks. So the lining of the uterus won't shed and you won't have your period. And actually HCG, human chorionic gonadotropin, is what is detected in a pregnancy test. Its highest is at about 7 weeks, I believe. And then this is sort of redundant, but HCG causes males to create testosterone or produce testosterone and tells your bipotential gonads to become testes and not ovaries. So this would be a male chromosome and then a female. So if it has this Y chromosome, then your bipotential gonads will become testes. Okay, thanks to this HCG. Okay. So once the embryo is embedded in the uterus, the corpus luteum lives for about 7 weeks, like we said. Then the placenta will take over, and the placenta is going to provide food, oxygen, CO2 exchange. And it's important to note that there is no actual mixing of blood. And it takes over progesterone production. Okay. So if I was to draw that for you, there is a fetal side and a maternal side for the placenta. So here's the wall of the uterus. Here is the fetal side, and then here is the maternal side of the placenta. And here's the blood supply. In red, taking food to the placenta, to the baby, and then taking clean blood to the baby, and then away, taking dirty blood away. And there are capillaries there that, and the blood doesn't mix. They are in close contact, but there's no mixing of blood. Okay. So then after this happens, we go through what's called embryogenesis. So you may remember this from lab. We started off with one cell called a zygote. And this is early embryogenesis. And it goes through what's called cleavage. And that is splitting without growth. This happens within the fallopian tube. So we really wouldn't want growth of our cell, or we wouldn't want growth of our embryo in the fallopian tube, because then our fallopian tube would explode. Remember, we also, surrounding our embryo, we have our zona pellucida here. It's also keeping the growth from happening. So we have cleavage, which is growth or splitting of our cells without growth. So we do this until we get to about 32 cells. So, for instance, there are about 32 cells inside here, and our zona pellucida is intact. Okay? And at this stage, it's called a morula, which is just a Greek word for mulberry. So embryogenesis is just the development of the embryo. Okay, and this right here, what we're looking at is early embryogenesis. And then those cells start to differentiate and become different things. So then this would be differentiation. So we have our cells inside here. Then the cells surrounding it start to become something else. And I'm doing that in different colors to represent that. The cells on the outside we term trophoblasts. And then on the inside we call embryoblasts. And they differentiate even more until we form a blastocyst. So we still have our zona pellucida here. And then our blastocyst our zona pellucida starts to form holes. And what do the cells on the inside start to look like? Well, they become even more compact. And migrate to one side. Our trophoblasts look like this. And we form this sort of space or cavity. This space or cavity is called a blastocele. And a lot of times they call the compacted cells, the compacted embryoblasts, the inner cell mass. Okay. Okay, so now we have holes in our zona pellucida. And we're ready for implantation. So we've been in our fallopian tube for 4 to 5 days. We make it into the uterus and ready to implant. We're going to bounce around in the uterus for about 5 days and implant to the side of the uterus. So then we go into what's called blastulization. And this happens after cleavage. And remember, the definition of cleavage is just splitting without growth. So we're not getting bigger, but we're becoming something different with cleavage. Okay? So what do our cells look like? Our trophoblasts here on the outside. These are our trophoblasts. Our inner cell mass. We formed another cavity in our inner cell mass called the amniotic cavity. And now our cells here in the middle have sort of formed this disc shape. So remember, this whole structure is spherical in nature. It's not 2D, it's 3D. And for instance, if we were to sort of chop this top part off, the amniotic part off, and draw our disc, they would look sort of like this. The top one would look like this, which is sort of like a pancake on top of a pancake. Okay? And they call this the bilaminar disc. Bilaminar disc. All right, the top cells are called, here in green, are called epiblasts. So upon, epi means upon, so these are epiblasts. And then the ones below it here in yellow would be hypoblasts. Hypo meaning below. Okay. So now let's take a section view. So if we pull these two pancakes out the bilaminar disc and draw it like this. So here are our two pancakes. And sort of take a sectional view of it. We could draw it, I'm going to draw it over here. Could draw it like this. Here are two sections of that. What happens is that we sort of get this formation of cells called the primitive streak. And it's like if we pour a streak of syrup down our top pancake in the epiblast. It's called the primitive streak. So that would be the primitive streak. And then that primitive streak, if we draw it here, remember we just took a section out of it, and we're looking at this section, and it's right here, just a part of it. What happens is that primitive streak, those cells start to migrate out into our bilaminar disc. Okay? And then our bilaminar disc goes through what's called gastrulation. And this is the forming of the germ layers. Okay. So now we no longer have a bilaminar disc, we have a trilaminar disc. Okay. Our top layer is called the ectoderm. The middle layer is called the mesoderm. And then the bottom layer is called the endoderm. And they will all differentiate and become the rest of our organs and so forth. So this trilaminar disc continues to differentiate and go through what's called neurulation. And you can sort of guess what that's going to be. So let's take a look at our trilaminar disc now. Our trilaminar disc, let's draw all the layers in. Here's our mesoderm. Our ectoderm here on top. And our endoderm on the bottom. And what begins to form is called a notochord in our mesoderm. This is a notochord. And it really doesn't do a whole lot, but cause a thickening in the ectoderm called the neural plate. So let's draw that in as a thickening in the ectoderm. This thickening is called the neural plate. Okay. Eventually, this notochord is going to zip up, or the neural plate is going to zip up. Show you what I mean. So here is our notochord and our neural plate starts to form this ring. And this ring is what is going to become the neural tube. You learned about this in A&P1. The neural tube will look something like this. And then you'll get a widening on one end, on the cranial end. This is the cranial end, and then this would be the caudal end towards the tail. And then eventually you'll get the formation of the 3 primary vesicles, and so forth. Okay. There are also other cells that differentiate out here that become neural crest cells that do other things in the body. And this becomes the nervous system. Okay. So this is early embryogenesis. Now let's talk about gestation, which is, of course, pregnancy. Okay. And I think it sort of helped to talk about the fact that they do this not only in months. So here I have it listed in white in months, and then in green in weeks. We'll also break it down into trimesters. Okay. So in this graph, we have hormone concentration, which we'll get to, and then it's broken down into time. All right. So we started it out at 0, and I think the beginning of gestation or pregnancy, they sort of start that at the last menstrual period, and they often term that as LMP, last menstrual period. And fertilization doesn't typically happen until about the 2nd week. So after you get pregnant, fertilization doesn't happen until about the 2nd week. Okay. And then after that, you have the zygote, and then you go through embryogenesis, and then you go through organogenesis, which is the development of the organs. And then after about 10 weeks, you have what's termed fetal development. So from 0 to 3 months is the first trimester. Okay. And then from 3 to 6 months is, of course, the 2nd trimester. And then from 6 to 9 months is the 3rd trimester. Once you get to about 24 weeks, you have what's called a 50% survival rate. 50% survival rate, which means that 50% of babies born premature have a 50% survival rate. But complications can become different and increase as well. Okay, so let's talk about hormones. What do the hormones have to do with this? So we have, our first one is HCG. Let me fix this curve a little bit. HCG tends to peak at about 7 weeks, because remember that's being produced by the corpus luteum that's in the ovary. And then after that, some of it is produced by the placenta, but it obviously will go down. Okay, so peaks at about 7 weeks. And then what about our estrogen and progesterone? We have peaks of those during the 3rd trimester. Okay, so estrogen and progesterone are increasing. They're making sure the uterus is hospitable for the baby, the fetus growing in there. But then we see a quick downfall during what would be termed parturition, which is the process of giving birth. The process of giving birth. And we see this downfall, or this drop in estrogen and progesterone, because remember, progesterone, it causes muscle relaxation of the uterus and prevents uterine contraction, which we will need to actually give birth. So of course we would see a drop in progesterone, and also estrogen helps in this as well. Okay, so preterm and postterm. When is preterm and when is postterm? At 40 weeks, anything past 40 weeks is considered postterm. And then anything, I think it's after 30, I want to say it's 36, is considered preterm. So on average, full term is like 38.5. This is full term. 38.5 is full term. Okay, or is that 37? Anyways, just know that full term is 38.5. I think that's all I really want you to know from that. The last thing I want to talk about is changes in mom. Okay, so estrogen. What is estrogen doing? Estrogen is suppressing, remember, FSH and LH, follicle stimulating hormone and luteinizing hormone. It's causing the growth of fetal organs. And it's increasing the growth of maternal tissue, like mammary glands. And the uterus. What about changes in the anterior pituitary? It causes actual growth of the pituitary gland. And growth of pituitary gland is going to stimulate increase in the thyroid gland. And in turn, this is going to increase metabolism, increase appetite, and also increase hot flashes. What about the circulatory system? All this growth, we're going to need an increase in blood volume, which it does, and a total of about 1 to 2 liters in increase. It will also increase blood pressure and pulse. What about the lungs? And all, they're going to work about 50% harder than normal. The stomach is going to push up on the diaphragm, causing things like gastric reflux, acid reflux. And what about weight gain? The fetus, on average, you're going to gain about 7 to 8 pounds. This is just an average, of course, from the fetus. The placenta is going to weigh about 6 pounds. from the breast, you're going to gain about 3 to 4 pounds. The uterus is going to gain about 4 pounds. From blood volume, you're going to gain about 4 pounds. And from total fat, they say anywhere from 3 to 10 pounds. So that means you're going to, on average, gain about 25 to 35 pounds during pregnancy. So, of course, this is just an average, but in total, about 25 to 35 pounds from all of this. Okay, so your final is coming up. Make sure to study your practice quizzes, go over my notes, and then just review over your Learn Smart. Remember, if it's not in the practice quiz or if it's not in my notes, you're probably not going to get asked it on the exam. Your final exam pulls questions from your previous unit exams as well, so you're probably not going to see new questions on the final. Okay? Best of luck to you. I hope that, and I know that you will do well. Remember to take your time, read questions carefully, and best of luck to you. Happy studying.